the different methods of pure culture techniques, right? So today also we are taking this an example. What are methods to isolate the bacteria from the given sample, isolation, enumeration, and pure culture techniques. So please confirm, are you able to see this slide, this board?
Yes, sir. Yes, sir.
Right. So on the last time we have discussed cultivation of bacteria, right? And we have discussed how to culture the bacteria from a pure culture, right? So last time हमने डिस्कस किया था कि एक प्योर कल्चर दिया हुआ था. प्योर मीन्स द ऑल द बैक्टीरियल colonies which was shown, that is, for example, E. coli, it was pure. It is known the population are of the same genera as well as the progenies of the same genera, and we have studied how to culture the bacteria, right, using different methodology, and we have discussed what are the requirements. Then we have discussed what is the difference between a medium and a culture, and how we do the inoculation, the process of inoculation. Then we have seen the incubation, concept of incubation, requirement of a particular temperature for the bacterial growth, how we'll observe the growth of the bacteria in a liquid medium, right, as it get turbid. Then we have discussed about the pure culture techniques, and for this we have discussed three plate method, four plate method, split plate methods, right? So today also we'll discuss how to isolate the bacteria from a given sample, right? Which is basically a mixed culture. So last time we have seen that suppose we have a mixed culture, how will isolate or get a pure culture, and the technique which is involved is pure culture technique. So now the thing is, what is mixed culture, right? So mixed culture is the culture which contains more than one type of bacterial population and its progeny. एक से ज़्यादा टाइप के बैक्टीरियल पॉपुलेशन, राइट, या उसकी प्रोजेनीज हो तो वो मिक्स्ड कल्चर कहलाएगा, यानी डिफरेंट बैक्टीरियल जेनरा इन्वॉल्व है. अब हमने एग्जांपल लिया था कि सॉइल सैंपल. Suppose we have provided with a soil sample. The soil is containing different type of bacterial population. We are talking about bacteria, not fungi or yeast or other microbes here. We are talking about bacteria, right? So the soil is containing different type of bacterial genera. How will isolate the bacterial genera from the mixed culture, and what are the methods involved? So serial dilution method, portrait method, spread plate method. So we discussed today. Right. So please again confirm me. Are you able to see this board written date 4th August 20 pure culture techniques? क्या आपको ये बोर्ड दिखाई दे रहा है जिसपे लिखा हुआ है प्योर कल्चर टेक्निक्स? आज की डेट डली हुई. Fine. Right. So we'll start with. Now let us suppose I'm taking with an example so that you can understand the concept. Let us suppose we have soil sample. Okay. Now idea is, or the objective is, to isolate the bacteria, from given soil sample, right? We are trying to understand the concept using practical approach, and definitely we do these experiments in the laboratory. Note point is here I have selected soil sample. The sample can be anything. Okay. So to isolate the bacteria from given soil sample, number one, then to enumerate, that is to count the bacteria, right, and then to isolate to get pure culture. Get pure culture. तीन ऑब्जेक्टिव्स हम लेके चल रहे हैं. पहले सॉइल सैंपल आपको दिया हुआ है और हम ये कह रहे हैं कि द सॉइल सैंपल इज कंटेनिंग अ डिफरेंट टाइप ऑफ बैक्टीरियल पॉपुलेशन, hence it's a mixed population. How we'll get the pure culture? So first we'll isolate the bacteria from the given soil sample, we'll enumerate or count the bacteria, and we'll how to get the pure culture. So first what we are doing, we are selecting the collection site from where you will get the soil. So it depends upon your interest, whether where you want to check the diversity of bacterial population, obviously in natural environment. So you will select the site, and from there you will get 1 gram soil sample जो कि dry होगा, wet नहीं लेंगे, and will weigh actually, and now we have 1 gram soil sample. Now we have already prepared test tubes. We'll take around 8 test tubes, right? तो अब हमने क्या किया कि टेस्ट ट्यूब्स प्रिपेयर की हुई हैं ऑलरेडी. प्रिपेयर का मतलब ये है कि आपने इसको स्टरलाइज कर लिया है यूजिंग ऑटोक्लेव. Okay, so I am labeling it 1, 2, 3, 4, 5, 6, 7, 8. 8 test tubes we have taken, and we'll take 9 ml suitable solvent. All are containing 9 ml distilled water or suitable buffer, right? So ये distilled water जो हम use कर रहे हैं, it is used as a solvent or any buffer you can take. Now the point is why I'm taking 8 test tubes. Can we take less than 8 or more than 8, right? So it basically depends upon the bacterial population in a soil sample. So it has been described that in soil sample, the idea is basically we have to dilute the soil sample, and what we are doing is we are putting 1 gram of soil in the first test tube. Now all the test tubes containing distilled water, सबसे पहले हमने क्या किया, test tubes लिया है, glass test tube you can take or plastic test tube you can take. We'll take 9 ml distilled water in each test tube, then we'll do the plugging. अब हमने plugging कर दिया इसमें, and after plugging we'll sterilize this first. We'll do the autoclaving at 121 degrees centigrade. यानी कि ये all the test tubes are sterile test tubes containing distilled water. These test tubes are called as blank test tubes or blank tubes. Blank tubes. Blank tubes means tubes containing solvent. Here it is distilled water. If it is not containing anything, we call it as empty tubes. Likewise, we use special type of plates. We call it as a micro-titer plates or 96 well plates. So you can use anything for dilution. If it is containing any solvent, then it is called as blank tubes. So here you have blank wells. So it is blank tubes means containing 9 ml of distilled water. Now point is again, why 9 ml? 9 ml distilled water क्यों लिया? इससे कम या ज़्यादा ले सकते हैं? So actually we are following a method which is known as serial dilution method, and it is 10-fold serial dilution method or technique. It is tenfold serial dilution method. This method is called as tenfold serial dilution method because here we will dilute the given sample tenfold, that is 10 times each step serially. We will serially dilute the given sample 10 times or tenfold at each step. Now see how. First, what we'll do, we'll take 1 gram of soil sample and resuspend in 9 ml of distilled water. Right? So this first test tube is suspended with 1 gram of soil sample. You can make even 1 ml of soil suspension. So here in the first test tube, right? Initially the distilled water was 9 and 1 gram of soil sample, 1 ml of soil suspension you are adding. The total volume is now 10 ml. So 1 upon 10. 1 is the sample, 1 part is the sample. In total volume is 10 part or 10 to the power minus 1. Right? So what you are doing at this step, you are diluting the soil sample 10 times. 1 part is the soil, any 1 gram or 1 ml soil, in total 10 ml of suspension, means 9 ml plus 1 gram or 1 ml, it becomes total 10 ml. So in 10 ml, 1 part is the soil sample. So 1 by 10 or 10 to the power minus 1, meaning 10 times. Now what you will do, you will add, you will mix, and after mixing it, then transfer this sample to the next tube 1 ml. 1 ml, this suspension will be transferred to the next test tube. And here again you will mix it. Mixing can be done by pipetting, right? By micropipette you can mix it. After mixing it, then again you will transfer 1 ml and likewise transferring, mixing, then transferring. So at each step you are transferring 1 ml likewise. And this you will discard. So you have taken 8 test tubes and you are doing the dilution of the soil sample serially. आप देख रहे हैं इस test tube से इस test tube में serially you are diluting the soil sample, and at each step you are diluting 10 times, because in this case in the test tube 2, right, the total volume of the distilled water is 9 ml and you are adding 1 ml which is already 1/10, 10 to the power minus 1 diluted. So here it becomes 1 upon 100 or 10 to the power minus 2. Likewise minus 3, minus 4, minus 5, 10 to the power minus 6, 10 to the power minus 7, 10 to the power minus 8. What is the principle? Principle is to dilute the soil sample containing bacterial population in such a way that it gets diluted 10 times at each step. And it has been studied that around 10 to the power minus 4 to 10 to the power minus 5 you will get the fungus, and at the dilution 10 to the power minus 7 and 10 to the power minus 8 you will get bacterial population, right? So you will get basically the isolated bacterial population at this dilution. So that's why we are taking 8. You can even take more test tube depending upon the population in the given sample. Right? So you have to standardize it actually. It is not fixed कि आपको इतनी test tubes लेनी है depending upon the population. But here in the case of soil sample, you will get the isolated colonies of bacteria 10 to the power minus 7 to 10 to the power minus 8 dilution. Now again, see what we have done. We have weighed 1 gram of soil sample. We have already prepared 8 or 9 test tube or 10 test tubes because you will take a control test tube as well, right? So all the test tubes are containing 9 ml distilled water, right? And you will autoclave it. After autoclaving, these test tubes are said to be blank tubes. You will use these test tube for the dilution of the soil sample. So we place all the test tube in a test tube holder or rack and label it 1, 2, 3, means from left to right, 1 to A, so that we can do the dilution, right, without any confusion. Okay, now the soil sample has been diluted. Then what you will do, say let us suppose I have selected 10 to the power minus 7 dilution. Now you will select this test tube or then and take diluted soil sample as 1 ml and 0.1 ml. That is 100 microliter. 1 ml or 100 microliter. So you have diluted the soil sample at 10 to the power minus 7, and you are taking 1 ml of the sample and 0.1 ml. Here you are also diluting 10 times more. So you are taking 100 microliter to assure that you will get isolated colonies. So what you have done, you have taken 1 ml of soil sample and 0.1 ml sample separately, and then you will pour it onto the media plate. So here you can do pouring, you can follow pour plate technique or spread plate technique, right? Pour plate technique or spread plate technique. मैंने आपको बताया था, in pour plate technique you will get aerobic and anaerobic both, right? And in spread plate technique you will follow, you will get aerobic bacteria only. So accordingly you will, so 1 ml of diluted 1 ml of what, 10 to the power minus 7 diluted 10 to the power minus 7 diluted soil sample. We're taking 1 ml and then spread it all over the media plate. This is the NAM plate. Same here, 0.1 ml, and you will spread it onto the plate. Right. So here you can follow spread plate method or pour plate method as per your requirement. Right? If it is asked to get both aerobic and anaerobic, you will follow pour plate. Right? आप pour plate technique follow कर रहे हैं if you want to get aerobic as well as anaerobic bacteria. लेकिन नोट यहाँ पर ये है कि if it is asked that isolate anaerobic bacteria, then for anaerobic bacteria you will follow anaerobic cultivation method. यानी इसका मतलब ये है कि apart from pour plate method, even though you are getting anaerobic bacteria, you will follow the protocol of anaerobic cultivation. That is actually different. That different means where there is no oxygen even in the medium or even in the incubation system. So notice what type of bacteria you are isolating. So let us suppose we are isolating aerobic bacteria only. What you have done? You have spread the sample 1 ml and 0.1 ml in the respective plates, and also you will take control media plate. जैसा हमने last time भी discuss किया था, you will also take control media plate to check the sterility of the process, right? And then you will incubate it at 37 degrees centigrade for overnight growth. तो आप एक बैक्टीरियल प्लेट को रख देंगे incubator में to get the bacterial growth at this temperature. So after incubation, you will get the colonies onto the plate. As I've already told you, since it was a mixed culture and there are different type of bacterial population, so you will get different colonies. मैंने आपको बताया था कि colonies will appear and show diversity in terms of size, in terms of color, in terms of texture, margin, etc. So I am showing here different type of colonies as indicator of different colors. Yeah, different color show कर रहा हूँ, right? Likewise here also you will get the colonies, but probably you will get lesser colonies as compared to the first plate because it was diluted more, 10 times more, and you will get comparatively lesser colonies. So you will observe the bacterial colonies. Now, as per the standard protocol or method, if the number of colonies is less than 30, it is said to be too few to count, and if it is more than 300, in somewhere it may be written 200, it is said to be too numerous to count. TFTC, TNTC. अगर colonies less than 30 हैं, तो we call it as a too few to count. बहुत ज़्यादा कम है counting के लिए. And if it is more than 300, we call it as a too numerous to count. बहुत ज़्यादा है count करने के लिए. Now, the colonies which is appeared onto the plate, you will count the colonies using colony counter. Colony counter. मैंने यहाँ पर लिख दिया colonies अगर less than 30 है या more than 300 है, तो colonies का मतलब count कर रहे हैं. और count करने के लिए जो मशीन यूज की जाती है माइक्रोबायोलॉजी लैब में, इट इज नोन एज colony counter. So colony counter is a machine or instrument which is used to count the colonies. It is basically a somewhat box-like structure. इस तरह का कुछ structure होता है box ऐसे. Okay, and यहाँ पर it is fitted with a lens. Here it has a lens. There are switches, right? And it is a transparent, basically a glass where you put the plate. यहाँ पर आप अपनी plate रख देते हैं जिसमें colonies है. Now you will observe over this lens. आप lens के ऊपर से observe करते हैं so that the colonies get magnified. And then we have a stylus or a pen, right, which is connected and which has a sensors like pen hai. And then when we now let us suppose this is the plate containing colonies. Now with the help of say same this stylus, when you put a pen over the colony, it shows one count. Here in the screen it will show one count. Then again you will put a pen over the colony, it will count 2. Likewise, you will count the total number of colonies, and actually it will count automatically, right? So say I have clicked this one, and it will count 2. Then I have put the stylus over the colony, so it will count 3. Or likewise, you will count. So this is colony counter, right? It may be fitted with a instrument or a system, right, where you can download that data. So this is how you are counting the colonies. Now, once you will count the colonies, right, then the next point is to check or to measure the बैक्टीरियल पॉपुलेशन इन ए गिवन सेल. और मेरा पहला जो ऑब्जेक्टिव था वो था आइसोलेट करना फ्रॉम द गिवन सॉइल सैंपल. वी हैव ऑलरेडी आइसोलेटेड इट. द सेकंड ऑब्जेक्टिव था टू इन्यूमरेट और टू काउंट द बैक्टीरियल पॉपुलेशन इन ए गिवन सॉइल सैंपल. So for this purpose, we'll use a formula which is known as CFU per ml. That is colony forming unit per ml. Equal to number of colonies into dilution factor upon volume taken. Please confirm me, are you able to this board? आपको बोर्ड दिखाई दे रहा है?
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir.
Okay. Now, the first objective was to isolate the bacteria. I mean, given now let us suppose sample can be water, can be anything. So I've taken soil sample. The first objective was to isolate the bacteria from the soil sample. Then we have isolated. Now we are calculating the number of population in terms of colony forming unit. Colony forming unit का मतलब है assuming that one bacteria as a unit is forming one colony. एक bacterial unit एक colony बना रहा है, तो कितनी ऐसी colony forming unit है per ml. एक ml में कितनी colony forming unit है, and the formula is number of colonies into dilution factor upon volume taken. अब यहाँ पर number of colonies क्या है? Here the number of colonies is the colonies what you have counted in the on the plate. Okay. Now let us suppose the number of colonies is say 180. Let us suppose number of colonies is 180. Number of colonies is 180. Then second is dilution factor. The dilution factor is the reciprocal of dilution. आप जब लेक्चर सुने तो आप नोटबुक लेके बैठे, and so that you can write. The dilution factor is the reciprocal of the dilution, and the dilution what we have used is 10 to the power minus 7. We have diluted a soil sample 10 to the power minus 1, minus 2, minus 3, minus 4, minus 5, likewise up to 10 to the power minus 8. Then we have selected 10 to the power minus 7 diluted sample. So here the dilution is 10 to the power minus 7 and thus the dilution factor, which is the reciprocal of the dilution. So here it comes 10 to the power 7 plus 7. So dilution factor is 10 to the power 7. So number of colonies is 180 into 10 to the power 7 upon volume taken. Volume is inoculum taken. So in first plate we have used 1 ml. In another we have used 0.1 ml. So 0.1 ml means आप 100 ऊपर इसको शिफ्ट कर देंगे. So it is 180 into 10 to the power 7 upon 1. That is 1.8 into 10 to the power 9. 1.8 into 10 to the power 9 colony forming unit per ml. Now it comes 1.8 into 10 to the power 9 colony forming unit per ml. So here we are talking about the total number of colonies, right? We'll definitely do this experiment in triplicate. यहाँ मैंने आपको एक plate show की थी. I've showed only one plate here, right? That we have used for this dilution. Yeah, यहाँ मैंने I've used one plate only, but you will use at least in duplicate, right? So that you can check the error. Or you can take the average of colonies. So colony forming unit per ml now becomes 1.8 into 10 to the power 9 CFU per ml. So this is how you will calculate the total number of vital population in a sample. So it can be asked as a numerical question. It can be asked like, say for example, the given soil sample or given sample has been diluted tenfold, right? It was diluted up to 10 to the power minus 10. 10 to the power 8 was selected for cultivation of the bacteria, right? And say the number of colonies is this. Calculate the colony forming unit per ml, right? So anything can be asked from this formula. Now next point is pure culture technique. So how we get the pure culture? Pure means single bacterial population. Now you have this colony and you have isolated colonies, right? Different colonies you have isolated, like this way. There are different type of colonies, right? It is containing different type of bacterial population, more than one type of colonies. Now you will follow the same method what we have discussed on the last lecture. Same method. Means what you will do? First you will select the colonies. How? You will do the screening. The screening of colonies, right? On the basis of the similarity. Let us suppose मैं जैसे मैंने दिखाया red color की colonies. So all these colonies are similar. Similar means same size, same color, texture. So you will mark it as 1. Likewise, then you are observing different type of colonies, and as मैंने यहाँ पर indicate किया red color से, you will label it 2 on the back of the plate, right? Likewise, you have green color 3, right? Likewise, you will label the colonies. You will screen the colonies on the basis of their different aspects, and you label it for further isolation. Now let us suppose this one, colony number 1, colony number 2, colony number 3, right? So what you will do? You will take all the different colonies in suitable tubes containing broth. ठीक है, अब आपने क्या किया, आपने एक से ज्यादा टेस्ट ट्यूब्स लिए हैं, depending upon the screen colony. You will take broth in each test tube. Obviously it will be the slice test tube, slice broth actually we can say, and we'll do the cultivation. The same method you will follow. You will use the inoculating loop, right? We'll pick the colony, we will pass it onto the broth and label the tube accordingly. Say for example 1, here it is 3, here it is 2, like that. Each separate colony will be selected and pass it onto the test tube containing broth. Right? After same thing, means mixing, you will incubate, that is incubation, say 37 degrees centigrade for overnight. Right? And then you subculture it or culture it onto the leaf. Now since you have selected single colonies, right, you can follow streak plate method. Okay, so please let me confirm again. Are you able to see and am I audible to you?
Yes, sir.
Thank you. So likewise, so what we are following, the quadrant streaking. टेक्निक जो क्लास लेक्चर में मैंने पढ़ाया था, सेम आप उसको देखेंगे आपको याद आ जाएगा. Same method what we are following. Now we have a mixed culture, a plate containing different type of population. Okay, different type of vector population. First we have screened the colony, then we picked each single colony and transferred it into the test tube containing broth. It is a sterile broth or sterile media. After inoculation and mixing and incubation, will streak it onto the plate. Now note point here is, after incubation you will observe turbidity, right? In each tube you will follow turbidity except the control tube, and this turbidity confirms the growth of the bacteria. ये confirm करेगा bacteria grow कर गया है. Now you are assured that okay, my test tube is containing the bacteria. Then you will transfer it onto the plate by method, streak plate method, that is quadrant streaking, right? And again incubate all the plates at 37 degrees centigrade. Again for overnight. And after incubation, you will get the growth of the bacteria onto the plate. Right? Now you will get colonies onto the plate, and obviously you will observe the colonies as per the streaking. You did the streaking, तो आपको colonies उसी तरह से दिखाई देंगी. So I am showing here black colonies, red colonies, right, and say green colonies, just to differentiate that you have isolated the different bacteria onto the different plates, and in each plate all the bacteria are same. यहाँ पर जो इस प्लेट पे जितने भी बैक्टीरिया है वो सब सेम है, क्योंकि आपने इसको प्रोपेगेट किया है, ग्रो किया है फ्रॉम द सिंगल कॉलोनी. एक सिंगल कॉलोनी को पिक करके आपने उसको प्रोपेगेट किया है ना, एक्सपेंड किया है. तो यहाँ पर उस कॉलोनी की प्रोजेनीज ग्रो कर गई हैं. सो इट विल द प्योर कल्चर, मीन्स It is containing single type of bacterial population as well as progeny here also, but it is unknown to you. अभी तक आपको ये जो कॉलोनीज मिली हैं, it is even it is pure, but it is unknown, right? It is unknown. Now again revise the things first. पहले हम revise करते हैं what we have done quickly. So we have taken soil sample, okay, and the soil sample you have diluted it tenfold. ये tenfold dilution किया. Then you have selected 10 to the power minus 7 dilution. From 10 to the power minus 7 dilution, you have selected 1 ml and 0.1 ml, right? Then you have spread it onto the plate. You have incubated it onto the plate. Then you will observe the colonies. With the help of colony counter, you will count the colonies. You will not consider those places on which the number of colonies is either less than 30 or more than 300. We have assumed that we have got one number of colonies 180. Then using the formula CFU per ml, number of colonies into dilution factor upon volume taken, will calculate colony forming unit. And here it is 1.8 into 10 to the power 9 CFU per ml. Now from the mixed plate or the plate containing different type of bacterial population, we'll do the screening. We'll select each colony. We'll mark, actually we'll label the colony, each colony. We'll transfer it to the respective test tube containing suitable prop. After proper incubation, each tube showing turbidity, that is overnight grown bacterial culture, in each test tube is then streaked onto the respective petri plates, right? Or here you should or you must follow the labeling method, यानी कि आपकी प्लेट ट्यूब की लेबलिंग प्रॉपर्ली होनी चाहिए to avoid mixing or confusion. After incubation, you will get the bacterial population onto the plate. Each plate will show a specific growth of bacterial population. It is said to be pure culture. Now you have isolated the bacteria, right, from a mixed culture in a pure form. And now onwards, next process will start, that is identification. Identification. और आपको याद होगा, even I have asked this question, which method of identification you have already studied. तो आपने बोला था staining, gram staining. So you will do the identification method. आपको colonies तो मिल गई, pure colonies भी मिल गई, but अब आपका काम है to identify the colonies, or identify मतलब to identify the genera. So you will do staining, right? Here staining will confirm you the gram positive property and gram negative property, and on the basis of cell wall. Then culture method, you have already studied culture method. आपको याद होगा मैंने बताया था selective cum differential media. So you will use selective cum differential media to get the colonies, to identify the colonies. To first identify whether it is gram positive, gram negative. If it is gram positive, which type of bacterial genera may be, and then you will follow biochemical tests. Biochemical test. ये बायोकेमिकल टेस्ट जो है, ये है ऐसे टेस्ट हैं जो कि बैक्टीरिया की बायोकेमिकल प्रॉपर्टी, बायोकेमिकल रिएक्शन बेस्ड है. जो बैक्टीरियल सेल में बायोकेमिस्ट्री रन हो रही है, यानी जब वो सबस्ट्रेट को यूटिलाइज कर रहा है और वो एंजाइम प्रोड्यूस कर रहा है, कौन सा एंजाइम प्रोड्यूस कर रहा है, प्रोडक्ट क्या बन रहा है, इस बेसिस पे डिफरेंट टाइप ऑफ बायोकेमिकल टेस्ट हैं. फॉर यू इंफॉर्मेशन, नाउअडेज high media kits are available, which are known as high media identification kit. And using these identification kit, you can easily identify the bacterial genera. ये कुछ इस तरह की किट्स आती हैं जो कि स्ट्रिप्स होती हैं और इसमें वेल्स होते हैं. इसके बारे में फिर मैं कभी और डिस्कस करूंगा इन डिटेल. And each well represent a there are different type of basically biochemical tests, right? So each will represent a different test, and it is basically a rapid test, and you can easily identify the bacterial genera. And then there are different methods like your immunology method or serological methods are there. आप serological method से identify कर सकते हैं या फिर ultimately you will follow molecular methods. Molecular methods जो आप molecular biology में डिटेल में पढ़ेंगे, यानी on the basis of their genetic diversity, genetic sequence that includes hybridization, sequencing, etc. Right? So till date, you have studied what is the basic nutritional requirement of bacteria, how the bacteria can be classified on the basis of nutritional requirement, what is culture media, what are different type of culture media, how to sterilize the media, then how to cultivate the bacteria in broth or on a solid medium. Then you have studied how to isolate the bacteria from the given sample, how to count the bacterial population, and how to get the pure culture from the mixed culture using pure culture technique, that is serial dilution method, steep plate method, spread plate method, board plate method, etc. Then how to identify, or a little bit about the identification. Is next step. Start with the identification. Finally, you identify the bacteria. Once you identify the bacteria and you have isolated the bacteria in a pure form, you will do the preservation or maintenance. You will maintain the bacteria, right? And this method is used to get the bacteria of your interest. इंटरेस्ट का मतलब बैक्टीरिया जो किसी स्पेसिफिक पर्पज से आप यूज करना चाहते हैं, किसी भी आप मेटाबोलाइट आइसोलेट करना चाहते हैं, राइट? आप किसी तरह की कोई खास स्क्रीनिंग कर रहे हैं बैक्टीरिया की फॉर स्पेसिफिक इंडस्ट्रियल एप्लीकेशन और पर्पस, यू विल फॉलो दिस मेथड. So this is all about the method of isolation and identification. If you have any query, you can raise your hand and you can ask me. आपकी कोई क्वेरी है, you can ask me. Yes.
सर जैसे हमको काउंट करना है अगर बैक्टीरिया को, तो हम उससे भी तो काउंट कर सकते हैं. पेट्रोल हाउसर काउंटिंग टेबल. उसको भी तो
No, we are actually counting the colonies, not the bacteria. Right? They're two different things. We are here counting the number of colonies, right? Colonies onto the plate. But if it is asked that a sample is given to you containing bacteria, count the bacteria, then you will follow different methods. बैक्टीरिया को काउंट करने के मेथड अलग होंगे, जिसमें स्लाइड मेथड होगा या अलग तरह के मेथड होंगे, डायरेक्ट मेथड या इनडायरेक्ट मेथड होंगे. बट यहाँ पर हम कॉलोनीज काउंट कर रहे हैं, राइट? एंड बेसिकली इट इज ए इनडायरेक्ट मेथड ऑफ enumerating bacterial population in terms of growth. अगर मैं कहूँ कि ग्रोथ को आइडेंटिफाई करें और डिफाइन करें या डिमॉन्स्ट्रेट करें, आप बताइए कि ग्रोथ हो रही है नहीं हो रही. आप सैंपल को लेंगे, इसको इनोकुलेट करेंगे, इंक्यूबेट करेंगे, राइट? और उसका ग्रो करेंगे. अब आप ये देखेंगे क्या बैक्टीरिया ग्रो कर रहा है, यानी कि बैक्टीरिया वायबल है. और फिर नंबर ऑफ कॉलोनीज कितनी है. तो दिस इज बेसिकली इनडायरेक्ट मेथड ऑफ काउंटिंग नंबर ऑफ cells. If it is asked direct counting or direct measurement, then we follow the slide methods. Yeah, anyone? Any other query you can ask? If you have any doubt, you can ask me. And suggest you to read practical book even more so that you can understand the practical concept, practical application of this basically the theoretical thing. Because ये जो आप units पढ़ रहे हैं ये सारे practical oriented पे हैं. Very interesting. You will find more information in practical labs. Any query? No? Okay, so probably this week we'll have a query session. इसी week में हम एक दिन query session करेंगे. You note down your queries and we'll discuss. Okay, thank you.